Alrighty, so this is going to be part 2 of Chapter 4's screencast PowerPoint presentation. And let me just move that over there. Okay, so where we ended, I was giving you an overview of how food molecules are broken down piece by piece, and as they are broken down, electrons pop off of them, and those represent the energy that is used to make ATP. So the processes to do this are stepwise, meaning that they're not all happening at once. They happen in a series of ordered steps, and those steps are 1, glycolysis, 2, the citric acid cycle, sometimes called the TCA cycle or the Krebs cycle. So if you see those terms on the internet as you're studying, just know that citric acid cycle, TCA cycle, and Krebs cycle are the same thing. And then finally, 3, oxidative phosphorylation. And in animal cells and in most organisms that have mitochondria, the final electron acceptor is oxygen. But as we'll talk about later, in some organisms it isn't. So regardless, we call this aerobic respiration, and it's interchangeable with the term cellular respiration. So this is not the same kind of respiration as breathing in and out, ventilating your lungs. It's different. Cellular respiration is actually the outcome of breathing in the oxygen that we breathe in. So if you've ever wondered why you need oxygen to live, I'm about to answer that question for you. So the reason that we breathe oxygen is for the following purposes. Okay, so electron carriers. So if we know that electrons represent energy, then if you're going to transfer energy, you need something to carry the energy around. So electron carriers are molecules that serve to transfer electrons from one molecule to another in the pursuit of forming ATP. So many of the electrons transferred between molecules do so as a hydrogen atom. So remember, here is what a hydrogen atom looks like. Here is a proton in the nucleus, and then a single electron in the valence shell of hydrogen. So if you need to carry around a wild, moving packet of energy in some kind of ordered way that's predictable, having it be in close relationship and proximity to a proton is a favorable way to do that. It's just an easy way to cart electrons around, just cart them as hydrogen. So in the processes that we're going to talk about here, transferring hydrogen, it really represents transferring of an electron. So, electron carrier NAD. So this is the most important electron carrier in ATP synthesis, and the word stands for nicotinamide adenine dinucleotide. So you don't need to memorize anything about the molecular formula of this. What you need to know about it is that it is the principal electron carrier in ATP synthesis, and it can be either empty or full. So if it's empty, it means it's lacking the electrons that it's carrying, and if it's full, it means it has all 2 electrons that it can possibly carry. So in its empty state, it looks like NAD+. So remember, anything that has a full plus charge, which means it is absent an electron. So lost an electron. Now, when you load it up, it becomes NADH. So you might be wondering, well, how do I go from an NAD+ to an NADH? Here's how. So NAD+, I should make that plus superscript. There we go. Plus 1 electron equals NAD. No longer plus because now the electron needs have been met. If I want to add another electron, I don't do this. That's not how it works. I instead just get the NAD to accept a hydrogen. So electron number 1 is here, and electron number 2 is here. So these electrons are then going to be carried by NADH to places where they can be used to create ATP. So that's the deal with the electron carrier NAD. So when you see that, note that this is the empty state, and this is full. So here's another view of the same idea, and in this one we have the NAD plus represented by a taxi. So the electrons are the people, and they're going to hail the NAD plus. The NADH is the fully loaded version, and the NAD plus returns when the passengers, the electrons, are dropped off. And here you can see a sort of stepwise or block style illustration down here. Oh no, the dreaded Microsoft PowerPoint pen. I want to use my pen. So note this part that I'm drawing a box around is NAD no plus. And if I want to convince this molecule to pick up an additional electron, I have to give it not just an electron, but an electron plus a proton, so a whole hydrogen. So this is electron number 2. And also it's important to note that reduced, if you are reducing something, it means you're sticking an electron to it. So if I am going to, oh, hello, Tiki. Tiki's near me on the couch. If I were to reduce Tiki, that would mean that I would be sticking an electron on her. By contrast, oxidizing is to take electrons away from things. So the reason this proton is said to be oxidized is because the NADH has stolen its electron. So that's the deal with that. So reduction and oxidation are two really common forms of chemical reactions, and as a group we call them redox reactions. And basically the way that we efficiently extract energy from food molecules in human and animal, and even just cellular biology in general, is through a series of redox reactions. So as electrons are picked up and then carried and then taken away and given to something else, you're getting a series of stepwise reductions and oxidations on the way to the final product. All right, so you need energy to think, to keep your heart beating, to play a sport, and to study this book. This energy is directly supplied by a nucleic acid called blah. So let's work through this. The answer is pretty obvious, but vitamins, we haven't discussed these, and also they're not nucleic acids. This is an electron carrier. This is a nucleic acid, and it's the energy dollar of the cell, so that seems like a good candidate. And enzymes are not nucleic acids at all. These are proteins, so those are automatically disqualified. So the only acceptable answer here is B, ATP. Okay, so I showed you that the process of transferring electrons to get energy out of them involves a couple different steps, and the first of those steps that I mentioned was something called glycolysis. So I'm going to make my little thingy smaller there. Okay, so glycolysis, and let's break down this word. Glyco means sugar, and lysis means break. So glycolysis is basically the first step in breaking down a sugar like a glucose molecule. So here we have glucose, which is a 6-carbon sugar. And the first step in glycolysis is that 2 ATP are spent. So these guys are going to be turned into 2 ADP. And the reason that we're spending energy is because we're going to break one of these bonds and straighten out this glucose. So it's going to go from being a ring to being a straightened out molecule of fructose with 2 phosphates glued to the end. So if I were to illustrate that, because they haven't, it would be C, 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 bound together, and then 2 phosphates on the ends, right? So once that's achieved, I've unraveled this circle and I've turned it into a line. Now I'm going to chop the line in half. So the outcome of the splitting of fructose diphosphate is 2 molecules of glyceraldehyde 3-phosphate. So glyceraldehyde is the name for this chemical. So it doesn't only have 3 carbons, it has other stuff going on with it too. It's just that for the sake of simplicity and ease of understanding, we are only illustrating the carbons in these molecules in this illustration. But there is, of course, hydrogens and oxygens and so forth all over the place, and that's what makes it glyceraldehyde. You can infer from the name that this is a glycerol plus an aldehyde group. And then 3-phosphate. What does 3 mean? Well, it means that the phosphate is on the third carbon. So 1, 2, 3, it's on carbon number 3. So that's where the phosphate's attached. All right, so glyceraldehyde 3-phosphate is present in 2 units, 1, 2, there's 2 of them. And then those undergo a series of enzymatic steps wherein first electrons are taken off of them and used to reduce NAD+. So this represents 2 electrons. This represents 2 electrons. And 2 ADP are phosphorylated to form 2 ATP. So this stands for adenosine diphosphate, which means it's an adenosine attached to 2 phosphate groups. If I want to turn it into adenosine triphosphate, all I have to do is glue an additional phosphate on there, which fortunately is possible given the glyceraldehyde 3-phosphate reaction substrate. So to phosphorylate means to add a phosphate. So all I have to do is add a phosphate to 2 of these, and I get 2 of these, 2 ATPs. So the end result is that you have 1, 2 NADH, and you have 2, 4 ATP, and you have 2 molecules of pyruvate, which is also called pyruvic acid. So if you see the words pyruvic acid and you're used to thinking of it as pyruvate, just be aware that those 2 terms are interchangeable and they mean the same thing. All right, so the summary for glycolysis, it's the first step in aerobic respiration. You chop up glucose in half and then do some stuff to it, and that results in 2 molecules of pyruvic acid. The net gain is 2 ATP, because remember, you gain 4 ATP total, but you had to spend 2 to split glucose, so if you do subtraction there, it equals 4. And you get 2 reduced NADHs, so those are going to have a total of 4 electrons between them, and those are going to be carried to the electron transport chain later. All right, so some additional notes on glycolysis. Glycolysis is a great unifying chemical reaction. It takes place in all living things. So anything that's a cell does this, be it bacteria, archaea, a tiger, you, your niece, whatever. Everyone does glycolysis. And it takes place specifically in the cytoplasm. So, oops, I hate this marker. There we go. So it takes place in the cytoplasm. And this is really important bullet point here because I expect you to know in which cell compartments things are going on. So it's the only form of energy harvesting in prokaryotes. So we call it anaerobic respiration. This means without oxygen. So glycolysis doesn't require the breathing of oxygen in order to work, and so prokaryotes don't require oxygen to perform glycolysis. It's the first stage of energy harvesting in higher level organisms, and then the reduced electron carriers are passed along to further steps in organisms that contain mitochondria, and they get sent to first the citric acid cycle and then later oxidative phosphorylation due to the electron transport chain. So glycolysis can be used exclusively, only glycolysis, not the second 2 steps, by eukaryotes when oxygen is limited. So for example, lactic acid fermentation is used by human skeletal muscles for short bursts of energy when we're exercising. So the rate at which your muscles use oxygen is quickly outpaces your ability to supply the oxygen, and that's when your muscles start burning, and that's when lactic acid starts building up due to the fact that your muscles have switched over to glycolysis instead. Okay, so here's the full version, and I know this looks really scary. I just want to remind you, you don't have to remember hexokinase, and you don't have to remember phosphoglucose isomerase, etc., so forth. You don't have to remember any of these enzymes that are catalyzing all these reactions. I'm really just trying to impress upon you how reliant cells are on enzymes to get their chemistry done. So that's the point we're trying to make here. So a different enzyme catalyzes each of these steps, and you can see the names of those enzymes in blue here. The name for this is a metabolic pathway, and I mean everything in this picture. So a metabolic pathway is the series of enzymatic steps that are required to turn one thing into another thing and release energy from it. So the goal is to pull energy out of the glucose molecule and use this to recharge ADP to ATP. All right, so quick thought question here. Glucose alone yields, in addition to 2 molecules of pyruvic acid, which of the following? Is it A, 2 ATP and 2 NADH, B, 2 ADP and 2 NAD+, or C, 6 ATP and 2 NADH? So if we go through this piece by piece, I automatically know that it's not B, because if the point is to extract electrons, NAD+ is not reduced, therefore no electrons were extracted whatsoever. 6 ATP molecules seems like a lot for just splitting a sugar, so I'm going to just eliminate that. And then A seems like a pretty good bet, so 2 total ATP molecules and 2 reduced electron carrier NADH molecules. So the only pitfall that you can potentially fall into here is that you could think, well, but at the end of that picture it said that there are 4 ATP, so it can't be this one. But remember, you have to spend 2 ATP to break glucose in the first place. You have to spend those ATPs to kind of straighten it out. So 4 minus 2 is 2. Therefore, A is the correct answer. Okay, so to transition between glycolysis and the citric acid cycle. So as we mentioned, glycolysis happens in the cytoplasm, but the business end of aerobic respiration occurs inside of the mitochondria. So what we have to do is look at how the pyruvic acid gets to the mitochondria. So pyruvate, as it's produced, builds up in the cytoplasm, and then it is translocated into the mitochondrion, but not between the membranes. So this right here is the intermembrane space. And that is not an ideal place for pyruvate to be. So in order for it to work, it has to be in the inner compartment of the mitochondrion, because that's where all the enzymes to perform the rest of these steps exist. So as you can see, pyruvate enters the inner compartment of the... Oh, excuse me. That one came on as a surprise, otherwise I would have paused for you. The inner compartment contains a series of enzymes that first take pyruvate and coenzyme A, coenzymes are things that help enzymes, this basically knocks a carbon off of the pyruvic acid and takes its place, which you can see right here. So we go from having pyruvic acid entering, and then acetyl-CoA is formed when coenzyme A kicks this CO2 off, and that also allows the reduction of 1 molecule of NADH, and that goes off to oxidative phosphorylation. The acetyl-CoA then enters the citric acid cycle and goes through a series of cyclical reactions to really get every last electron off of that sucker that's possible. So here's what that looks like. The citric acid cycle / Krebs cycle / TCA cycle looks like the following. We have pyruvic acid, aka pyruvate, and it's a 3-carbon molecule, but that last carbon, so this one, is going to exit in the form of CO2, and taking its place will be coenzyme A. So it's going to stick in the place where the carbon used to be. During that time, you get 2 electrons coming off, and those are used to reduce NADH. So now this can go off to oxidative phosphorylation, to the electron transport chain. Acetyl-CoA gets sent into the citric acid cycle, where the first step is that coenzyme A pops off again, and then each of these steps here is catalyzed by an enzyme, and each molecule of acetyl-CoA goes through this whole thing, or excuse me, each molecule of glucose, acetyl-CoA goes through twice. So the net products of the citric acid cycle per molecule of glucose are 6 molecules of reduced NADH, 2 molecules of FADH2. This is similar to NADH. It's just a different electron carrier. It's slightly less common as well. And then 2 molecules of ATP. So you can see that there's only 1 being produced here, and there's only 3 produced here. So this whole thing goes around twice. So in the citric acid cycle, derivatives of the glucose molecule in the form of pyruvate and acetyl-CoA continue to be broken down. So the cycle turns twice per glucose, and the result is that more energetic electrons are transported by the electron carriers over to oxidative phosphorylation. And you get the direct input of 2 ATP. So between glycolysis and the citric acid cycle, you get a total of 4 ATP, which is more than you had before, but not very much, all things considered. So this is the full version of the Krebs cycle, and once again, I don't expect you to memorize the enzymes and the steps and the products and the intermediates. I'm just showing you a metabolic pathway, and I'm showing you that enzymes are responsible for making these steps happen. So without enzymes, none of this would be possible. So the goal is still to pull energy out of the derivatives of the original glucose molecule, and use this energy to recharge ADP to ATP. So blank and blank are important, not so much for the ATP produced in them, but for the yield of electrons, which are picked up primarily by NAD+, that are used in blank. So this is a complicated one. You're going to have to think syntactically, so in terms of sentence order. So blank and blank, they are important not so much for the ATP produced in them. Well, that kind of tells me that the amount of ATP that they're making is maybe not that impressive. So glycolysis is a good candidate because it has a net yield of 2, but what else has a low yield? The citric acid cycle. So the only choice that has glycolysis and the citric acid cycle in the first 2 positions is choice C. And oxidative phosphorylation is the ultimate destination of the reduced electron carriers also. So this is a really good candidate answer, and in fact it is the correct one. So these and these are not correct. So I just gave you an example of a mental checklist or walkthrough that you could do during a test to narrow down what particular answers are. Okay, so oxidative phosphorylation. Let's actually break down this word now, because remember, it's always helpful if we break down big scary words into smaller chunks. So remember, to oxidize something is to take an electron away from it. So reducing is giving of electrons and oxidative is taking away. So and phosphorylation is to add a phosphate group to something. So the oxidation part is performed by enzymes in the electron transport chain, and that's just a way to pass electrons along to meet a particular goal. And then the goal is really to create a proton gradient in the intermembrane space, and that permits the phosphorylation of ADP to ATP. So this occurs on a series of molecules located on the inner mitochondrial membrane. So not here and not in here, but on this membrane in particular. So let's look a little closer at that. So now that we've defined oxidative phosphorylation, we have to talk about what is happening there. So previously, we've been looking at electron carriers picking up electrons from various glucose intermediates and then going away, doing something with them. The something is that they are taking their electrons over to something called the electron transport chain. So ATP gets synthesized by the energy released as the electrons flow through the protein molecules of the electron transport chain. And that's even a little reductive, as I'll explain in a moment when we get to that slide, so just stay tuned for more on that. The last electron acceptor in oxidative phosphorylation is oxygen. Oxygen accepts the electrons and in the process forms water. Okay, so here is the electron transport chain. Now all of these enzymes and coenzymes that I'm making little pink marks on, all of these guys, they all have names. And their names are things like cytochrome C oxidase, etc. But I'm not going to expect you to memorize those names. I just need you to kind of understand the gist of how it works. So NADH is coming over here, and it's going to pass its electron to the first enzyme in this chain. So they're numbered because they go in order. This electron is going to be passed within the membrane protein complex, so it's never leaving and it's never going down here. It's staying sort of in this zone that I'm tracing with my pink marker. And what that's doing is there's lots of protons in here. Some of them even come from the NADH themselves. So these protons are being pumped against their concentration gradient. So the actual reality is, in the intermembrane space, there are a lot of protons. All right, that's a little bit more reflective of the space between the inner and outer membranes. So the thing is, there are fewer protons in here, and there are more out here, which means that to move protons from in here, where there are fewer, to out here, where there are lots, that's pumping something against a concentration gradient, right? So we're going from an area of low concentration to an area of high concentration. And if you've studied active transport well enough, you would know that what that really means is you need to spend ATP to make this possible. So some energy has to be invested in order to get those proteins across up their concentration gradient, because it's not an energetically favorable way for them to move. However, we have a problem here. So the whole point of this endeavor is to create ATP, so to phosphorylate ADP and turn it into ATP and use that for energy. So it doesn't really make sense to pump protons across the membrane into the intermembrane space using ATP, because then you'd be spending it as fast as you were creating it. So there's an ingenious mechanism that works here to pump protons across without spending ATP, and here's how it works. The electron that is submitted to the electron transport chain via NADH is used as an attractant to protons. So remember, protons are a H+ ion, and they have a full positive charge, and electrons are high energy units that have a full negative charge. A proton really, really wants to get at least one, if not possibly two electrons to have its valence needs met. And it's going to be very attractive to electrons. So if you take an electron and put it in this system, protons are going to approach because they're going to be attracted to this electron. So this proton right here might be sort of looking at or sensing this electron and attracted to it. And maybe this one is too. By attracting protons to this area, protons can be sucked up into these proteins, these enzymes. And here's the way this works, and I'm actually going to erase some of my marks here so I can really show you explicitly. The proton ends up moving this way towards the electron that's presumably like right here. At the last second, the electron is whisked away to the next carrier in the chain. By that time, the proton is already inside here, so it just continues on through because it has momentum at this point. And that's how it works for the rest of the chain as well. Electrons are used to lure protons into the enzymes of the electron transport chain. And then at the last minute, before the electron and the proton get to meet, the electron is whisked away to the next thing, and the proton, having momentum of movement, moves on through. So that's the point of all of those reduced electron carriers, is to supply a constant stream of electrons to this process. Now, molecular oxygen, which we get by breathing, is sitting over by the last enzyme. And the final electron acceptor, it accepts 2 electrons and becomes water through a chemical reaction that you don't need to understand. But notice that final electrons plus O2 yields water, and this is why we need oxygen and also why we breathe out water. So this is called metabolic water. And that just means it's the water that we make as a consequence of metabolism rather than water that we consume to hydrate ourselves. So Over here, we have ATP synthase. I'm going to trash everything I just drew. So this takes advantage of that proton gradient. So as protons want to flow back down their concentration gradient, they don't have any option but to do so through this ATP synthase because there's no other proton transporter available. They can't go back through this, so they can only go through here. As the protons flow down their concentration gradient through the channel in ATP synthase, this molecule rotates like the turbine on a dam. And every time it rotates, the kinetic energy of rotation is sufficient to phosphorylate one ADP to an ATP. So for every glucose molecule spent between the ones produced by glycolysis and by the citric acid cycle and the ones produced here, you get about 34 ATP per glucose molecule. So oxidative phosphorylation is obviously producing the bulk of ATP of all of the steps. So it's really, really efficient at making ATP. It's better than the other two steps by far. Okay, so the electron transport chain is a set of molecules that supports a series of oxidation-reduction reactions. Remember when I said redox reactions are ones in which electrons are being either given or taken away? That is a redox reaction. So when the first enzyme in the chain passes its electron to the second, it is being oxidized. It's having an electron removed from it, and the next acceptor of the electron is being reduced. So that's what that means. ATP synthase is the complex molecular enzyme that uses that proton gradient to regenerate ATP from ADP. So we call this chemiosmosis, and this just is a word that means the potential energy provided by that proton gradient across the membrane is converted to kinetic energy when ATP synthase spins around, and that kinetic energy is converted into chemical energy, which is stored in the outermost phosphate bond of ATP. So that's all stuff I just told you. It's just if you like it in words style, here you go. Okay. Aerobic respiration. This requires mitochondria and it requires oxygen. And not all organisms that are alive have mitochondria or live in places where oxygen is available. So in those cases, something else needs to accept electrons from NADH instead, because otherwise, why would you make NADH? It has no intrinsic value by itself. Its only use is to pass electrons around. So we call this anaerobic respiration, which means without oxygen. And this can occur in many forms. So one form that I kind of obliquely mentioned earlier was the idea that for some organisms there isn't oxygen around, so they use sulfate or methane or nitrate as the final electron acceptor. So these tend to be archaea or bacteria that make stinky byproducts that are stinky. Lactic acid fermentation is one form, and that's the form that occurs in animal cells when they run out of oxygen. And finally, ethanol fermentation. So this is the one that produces products like beer and wine that people like to drink, but they're really just the metabolic outcome of fermenters. So here's an example of oxygen replacement. So you can see this green color here in these coastal waters. So this spooky bloom of algae. And then here you can see it taking the form of the waves that are crashing against the coast. These are from an eruption of hydrogen sulfide. So hydrogen sulfide is H2S. So anaerobic sulfate-reducing bacteria release hydrogen. So sulfate-reducing means sulfate is the final electron receptor. So sulfate is SO4 minus. And for this guy, the final electron acceptor is sulfate. And what they give off as a byproduct after the electrons are accepted is hydrogen sulfide gas as they decompose algae in the water. So they're breathing sulfur rather than oxygen. And ATP is made directly from the NADH electrons that are used in this process. Okay, lactic acid fermentation. This is common in mussels when they have become exhausted by use. So if you are running or working out so hard that your muscles are using oxygen faster than your lungs can supply it, the muscles will switch over to lactic acid fermentation, and that's how you get sore. So the idea in lactic acid fermentation, all the steps of glycolysis are the same. But instead of having the 2-pyruvate go into the mitochondrion, they stay in the cytoplasm and undergo fermentation to become 2-lactate. So lactate and lactic acid are the same thing. But, so here we see we're reducing 2-NAD+ to 2-NADH. And here we are turning 2-NADH into 2-NAD+ in order to do the fermentation. What this does is keeps a constant supply of NAD+ going so that the 2 ATP produced by glycolysis can happen again. So it's not a very efficient way to produce ATP because its yield is really low compared to that of oxidative phosphorylation, but it does create smaller amounts of ATP faster. So that's why I call it the quick and dirty way to make ATP. So it's fast, it's not as efficient, and it creates a nasty byproduct that can hurt your muscles if it builds up too much. Okay, ethanol fermentation. So this one takes pyruvic acid, so the product of glycolysis. So again, all the steps prior are the same. And those are converted to carbon dioxide and acetaldehyde, which is an alcohol precursor. So acetaldehyde is taken and it is converted into ethanol by having 2 electrons removed from it. So acetaldehyde looks like this, ethanol looks like this, and it has an OH group at the end, which is why it's an alcohol. And this alcohol is the kind that humans like to drink. So things that humans have used ethanol fermentation before, and these come in forms of certain kinds of yeasts that are used to brew beer and wine, etc. So beer, wine, mead, soju, which is a Korean rice wine, rice alcohol, etc., sake, so on and so forth. So any alcoholic beverage that humans have discovered that they enjoy imbibing is produced via this process. Now, the nuanced flavors and such of those are produced from the grains that they are used as food for the fermenters and how long they're fermented and under what conditions, etc. But regardless, this is the chemical reaction of interest for that particular pursuit. So one job that you can totally have is to be a microbiologist for a brewery because they rely on microbes to make their alcohol. Okay, so we've been talking about carbohydrates as the primary substrate for all this stuff, but the reality is your body can use different molecules for fuel. So it's not kind of one size fits all, glucose only until the end, but rather it's just that the body and animal cells or cells with mitochondria even, they just prefer polysaccharides, so carbohydrates as fuel. It's not the only thing they can use. So if we look at this little chart here, at the bottom, it walks us through the steps. We know that glycolysis is step number 1. Pyruvate oxidation is step number 2. Citric acid cycle is step number 3. And finally, oxidative phosphorylation is step number 4. So, carbohydrates are the primary substrate that starts here, as well as some amino acids and, of course, glycerol, because it's almost exactly half of a glucose. But if the body needs to rely on other fuels, it can use fatty acids from the triglycerides stored in our fat cells, but those don't start at glycolysis. They go straight to this, and also some amino acids as well. Other amino acids start and can be fed into the citric acid cycle, but the products of all of these things end up with oxidative phosphorylation. So the order of operations for fuel use in the body is, excuse me, I just tried to scratch my nose and bumped the mic. Carbs first, fat second, protein third. And that, my friends, is the end of chapter 4. So your upcoming exam covers chapters 3 and 4. So cells, membrane transport, and energy uses, including the stuff we just talked about. So I wish you the best of luck on your exam and look forward to seeing you this week. Thanks for your attention and have a good day.